a beginner's guide for 16S, 18S, and ITS Amplicon Metagenomic Sequencing. So I would like to break today's presentation into 4 parts. Firstly, is a brief introduction of what it is of an amplicon sequencing. Then the project workflow from DNA extraction to bioinformatic analysis. And after that, we will have a led discussion, or we could call it a comparison among NGS 16S amplicon sequencing, shotgun metagenomics, and full-length 16S amplicon sequencing by using the PacBio platform. And you may find my assistant today, Dr. Novo, popping around the slides. So first, let's go back to last century, 1991, when environmental genomics was firstly brought to the world by Dr. Pace. And it is based on the sequencing of amplicons, or we can call it PCR products, of ribosomal rRNA genes. And the major purpose of amplicon metagenomic sequencing is to reflect the composition of microbial diversity in each various system. And in most of the cases, amplicon sequencing can be used to study diversity of bacterium, fungi, and some eukaryotic microbes in this community. And in 16S, 18S, or ITS amplicon sequencing, the target rDNAs, as is shown here on this slide, composed of conservative regions, which are labeled gray, and they are interspaced by the hypervariable regions, which are labeled blue and green here. And those hypervariable regions can provide species-specific signature sequences, which will be very useful for the identification of bacteria, fungi, and etc. For example, V4 region of 16S rDNA is the most common target region for sequencing in order to identify bacteria. What we usually do in amplicon sequencing is that we use universal primers that have binding sites on the conservative regions to amplify the target hypervariable regions out. And then we conduct the bioinformatic analysis to decide the phylogeny. And after some basic introduction, let's take a look at the project workflow of amplicon sequencing. At Novogene, we provide the entire solution for 16S, 18S, or ITS amplicon metagenomic sequencing, which means you give us your samples, we will return with sequencing data and bioinformatic analysis results. So when the sample first arrives, our lab technicians will perform a DNA extraction by using different kits or methods based on the type of your samples. And then after sample QC, we will use specific primers to amplify the target region of interest and do purification by using this magnetic beads. And after that, the qubit fluorometer will help to quantify the PCR products. And we will use agarose gel electrophoresis to separate and recover the target fragments. The next step will be library preparation. And finally, we will take the library to NovaSeq for pair N 250 base pair sequencing. For those of you who would like to send us genomic DNA, which means you would do extraction on your own. And we also have several kits or method recommendations. The first ones are the classic surfactants and ethanol precipitation approaches like CTAP and SDS. And also we could replace ethanol precipitation with magnetic silica beads. And the last one, this kit, the catalog number we have here, 51704 from Kaigen, is also a great option for doing extraction of DNA from those types of samples. And after DNA extraction, we will move to sample quality control. So for sample QC, we have mainly three criteria. The first one, we could look at the picture to the left here, that in which the main band is tight and can be clearly observed, and there is no obvious degradation. Criteria number two is that the DNA should be free of protein contamination. We may look at the picture here to the right that we can see the bands up here in the loading well, which is basically the protein contamination. We don't want that. And the third criteria is about transparency. We should observe no obvious color, and the sample should be totally transparent. For example, we could see yellow and red in samples in the picture number 2 and number 3, which are samples of abnormal transparency. And this indicates that there might be some problem with the samples that could probably sabotage our subsequent processes. And in this case, we will continue the process only after we got confirmation from you. And generally speaking, however, we do not recommend to continue. Instead, we would suggest you resending the samples for a better experimental result. And after sample QC, we will move to PCR amplification and library separation. So first, when we got the extracted genomic DNA, we will first fragmentize them, perform end repair, phosphorylation, A-tailing, and then we will add in specific primers which are linked with different barcodes to go to PCR amplification. And after PCR is done, we use qubit fluorometer for quantification and the agarose gel electrophoresis to recover the target amplicon fragments to move on. And the next step, we ligate the amplicons with the indexed Illumina adapters to go sequencing. So in 16S, 18S, or ITS amplicon sequencing, NovaSeq 6000 is used for this service. And the strategy we apply is the pair and 250 base pairs. And after sequencing, we will move to the data quality control. Here we label E as the sequence error rate, then Q fret, which is the fret quality, can be written as -10 multiplied by the logarithm of E with base 10. So if fret quality or fret score is 30, which means the error rate is 1 of 1,000, and then the correct rate will be 99.9%. We call this Q30. And although the sequencing quality will drop during sequencing, or the error rate here will increase due to the consumption of the reagents, we are still able to say that in most of the cases, more than 80% of the reads could reach Q30, which exceeds the data quality of Illumina's official guarantee of 75%. So for data quality control, one of the important steps is reads filtering. And what we do is that we will filter out three kinds of reads. The first one is that the reads contains a whole or part of the adapters. And the second is that is a read that contains more than 10% of bases that are uncertain after sequencing. And the third one is the read that more than 50% of bases with fret quality score less than 5. So after these filtrations, we will get clean reads for subsequent analysis. And at Novogene, all read sequences are stored in FASTQ formats. And we will include this pie chart in this data QC report, which will show the percentage of different kind of reads that I have mentioned above. After data QC, if you chose the bioinformatics package, Novogene, and we would continue to the analysis part. So firstly, using the clean reads obtained, we will do OTU clustering to categorize reads into different OTUs. And then the species annotation, alpha diversity, which is the diversity within samples, by the way, and the beta diversity, which is the diversity among different samples. And all of those analyses are listed here will be conducted. And of course, aside from the analysis items here, we are also able to do some advanced analysis like the variance statistics, the correlation analysis, the function prediction using the clean reads we just got. And in the next part, I will walk you through these analyses individually. So first and the most essential one is the OTU identification. OTU, namely the operational taxonomic unit, is determined with a cutoff value of similarity by the sequences of rDNA we got. So generally, this cutoff value is 97%, which means if the reads share 97% of the sequence similarity, they will be considered from one taxonomic level. And in the process of constructing OTUs, the basic information of different samples was collected, such as text data, text reads that went through splicing after sequencing. And the summarization is shown as in this figure. The Y1 axis here to the left is the number of tags, and x-axis is the sample name, and the Y2 axis titled OTU numbers means the number of OTUs which displayed in this purple bar. You can see here is purple bar to identify the numbers of OTU in different samples. And the red bars or total tags means the number of effective tags we got. And the blue bars or taxon tags means the number of annotated tags. And green bars or unclassified tags means the number of unannotated tags. And orange bars or unique tags, as we see here, means the number of tags with a frequency of 1 and only occur in one sample. In order to quickly and intuitively display the microbial community composition and abundance information in the samples, we would construct this taxonomic annotation corresponding to OTUs, and the results could be viewed in this heat map. So here in the filter above, we could input count value per OTU. When this value is decided, we keep the filter value unchanged and click this sample ID button. Then the graphic will be generated as the figure below. It will show the different taxonomy and the sample names and the corresponding OTU ID to the right. The counts are colored based on the contribution percentage of each OTU to the total OTU count in one sample. So here, the blue box contributes a low percentage of OTUs to sample, and while the red ones means they contribute higher percentage of OTUs. And according to the analysis result of OTU clustering and some of our research requirements, we normalize the OTU table and analyze both the common and unique information of different samples or groups. Then we can generate the vein or flower diagram. And each circle or petal represents one sample or one group, and values in the overlapping parts represents common OTUs, and the other parts are specific OTUs in each sample. So for the sake of artistic, we suggest to use Venn diagram if samples or group number is no more than 5. And if we have more than 5 groups or samples, using flower diagram will be much clearer to see. Then we show the high relative abundance genera to make taxonomy tree by independent R&D software. And we can do taxonomy tree either in samples or in groups. In this taxonomy tree here, the different colors represent different taxonomic ranks. The size of the circles, the relative abundance of species. So the bigger the size, the higher the abundance. And the first number below the taxonomic name represents the percentage in the whole taxon, while the second number represents the percentage in the selected taxon. Well, Corona display is an interactive metagenomic visualization in the web browser. And Corona visually displays the analysis results of taxonomic annotation. And circles from inside to outside stand for different taxonomic ranks. And the area of sectors means respective proportion of different OTU annotation results. And according to the taxonomic annotation results, we select the top 10 taxa of each sample or group at each taxonomic rank, that is phylum, class, order, family, or genus, to form the distribution histogram of relative abundance of taxa. So we could visually see the taxa with higher relative abundance and their proportion in different classification levels of each sample. The relative abundance of taxa in this phylum is illustrated in these two figures above. So the y-axis represents the relative abundance and the x-axis represents the sample name, and others here represents a total relative abundance of the rest phyla besides the top 10 phyla shown in this picture. And according to abundance information of top 35 genus of all samples, the heat map can be drawn so as to check whether the samples with similar processing are clustered or not. And the similarity and difference of samples can also be observed. And the results will be shown in this heat map here, these two figures. So plotted by sample name on the x-axis and the y-axis represent the genus. Here, the absolute value of z represents the distance between the raw score and the mean of the standard deviation. That means the redder the color here in this little box, the more this phylum clustered in the sample, and vice versa. And in order to find the differences in dominant taxa among three groups or samples that the selected taxonomic rank, we use the top 10 taxa to generate this ternary plot on the left. So the three vertices represent three samples or groups, and the circles inside represents dominant taxa, and the size of circle are proportional to the relative abundance. That means the higher the relative abundance, the bigger the circle. And the sample or group which the circle close to has higher abundance of this phylum. And also to further study the phylogenetic relationship of genus, we select top 100 genera, and the evolution tree can be drawn using the aligned representative sequences. So the relative abundance of each genus was also displayed along the genus in this figure. And the different colors of the branches represent the different phyla, while the different color represents different groups, and relative abundance of each genus in each group was displayed outside this circle. And alpha diversity is applied to the analysis of microbial community diversity within the sample. By analyzing the diversity of the single sample, we can reflect the richness and diversity of microbial communities in each sample, including biodiversity curves and a series of statistical analysis. And generally speaking, OTUs generated at 97% sequence identity are considered to be homologous in species, and statistical indices of alpha diversity when the clustering threshold is 97% summarized at this table. Here, this figure to the left is the rarefaction curve, and this is the rank abundance curve. So those curves are widely used for indicating the biodiversity of the samples. The rarefaction curve is created by randomly selecting a certain amount of sequencing data from the samples. then counting the number of the species they represent. So in this rarefaction curve, each curve represents a sample and can be colored and shaped by each sample name supplied in the mapping file. And the sequencing number is on the x-axis, and the observed OTU number is on the y-axis. The rarefaction curve can directly reflect the rationality of the sequencing data volume and indirectly reflect the richness of microbial community in the samples. So if the curve is very steep, here, if the curve is steep, a lot of species remain to be discovered. And if the curves becomes flatter, that means a credible number of samples have been taken and that only the scarce species remain to be sampled. To the right, this rank abundance curve is to sort the OTUs in the sample by the relative abundance from the largest to smallest to get the corresponding sorting number. And then we use this OTU sorting number as the x-axis. The relative abundance in the OTU will be the y-axis. And we connect this point with polylines to obtain this rank abundance curve, which can directly reflect the richness and evenness of species in the samples. So for the rank abundance curves, each curve represents a single sample, which can be colored and shaped by each sample name supplied in our mapping file. And in the horizontal direction, the richness and the richness of the species, is reflected by the width of the curve. So the higher the richness of the species, the greater the span of the curve on the horizontal axis. And in the vertical direction, the smoothness of the curve reflects the uniformity of the species in the sample degree. So the smoother the curve, the more uniform the distribution of species. And beta diversity represents the explicit comparison of microbial communities based on their composition. To compare microbial communities between each pair of community samples, a square matrix of distance, or we call it dissimilarity, was calculated to reflect this dissimilarity between the certain samples. The data in this distance matrix can be visualized with principal coordinate analysis, that is the PCOA, or principal component analysis, which is the PCA, the non-metric multidimensional scaling, which is the NMDS, and the unweighted pair group method with arithmetic means, which is the UPIGMA. So weighted unifrac distance and unweighted unifrac distance were selected to measure the dissimilarity coefficient between pairwise samples, which is a widely used phylogenetic measurement method in microbial community sequencing projects. And the heat map based on the weighted unifrac and unweighted unifrac distance is plotted here. So if we zoom in, each grid represents a pairwise similarity coefficient between the pairwise samples. And weighted unifrac distance displayed above, and unweighted unifrac distance is shown below. So the smaller the number shown here means the smaller the species diversity difference between the paired samples. And principal coordinate analysis, which is the PCOA, is an ordination technique which picks up the main elements and structures from reduced multidimensional data series of eigenvalues and eigenvectors. The technique has the advantage over PCA that each ecological distance can be investigated. Also, weighted unifrac and unweighted unifrac are calculated to assist this PCO analysis. So in this figure, each point represents a sample plotted by a principal component on the x-axis and another principal component on the y-axis. And the points were colored by group. So the percentage on each axis indicates the contribution value to discrepancy among samples. That is, it's the more similar the sample composition, the closer they will be to each other. And non-metric multi-dimensional scaling analysis, which is an MDS, is a ranking method applicable to ecological research. It's a nonlinear model designed for a better presentation of this nonlinear biological data structure, which aims at overcoming the flaws in methods based on linear model, including PCOA and PCA. So each data in this NMDS graph stands for a sample. The distance between data, between these data points reflects the extent of variation. The samples belong to the same group in the same color, as we can see here. And when the value of stress factor is this value on the up right side, when this value is less than 0.2, it is considered that this NMDS plot is reliable. Also, to study the similarity among different samples, we can use a clustering analysis to construct a cluster tree. The unweighted pair group method with arithmetic mean, which is the UPIGMA, is a hierarchical clustering method that widely used in ecology for the classification of samples. And the basic idea of UPIGMA are first, samples with the closest distance are clustered together to form a new node as a new sample, and its branching point is 1 second away from the original 2 samples. Then the average distance between the new created sample and the other samples is calculated, and the 2 closest samples could be found again to repeat the above steps. So a complete clustering tree could be obtained until all samples are clustered together. That is shown here to the left. But also weighted unit distance matrix and unweighted matrix were calculated before used for this UPIGMA clustering analysis. So here you could see that UPIGMA tree on the left and this relative abundance in phylum map on the right. And let's see, that is the linear discriminant analysis, this LDA effect size was used to analyze and detect biomarkers. So LabSea is a software designed to discover high-dimensional biomarkers and reveal metagenomic characteristics that including genes, metabolites, or taxa. So it can be used to distinguish 2 or more biological classes. And histogram of the LDA scores and cladogram are shown as a result of LabSea analysis for evaluation of biomarkers with a statistically difference among different groups. The histogram of the LDA scores represents species or biomarkers whose abundance shows significant differences among groups. So the selecting criteria is that the LDA score are larger than the set threshold, which the default number is 4. So the length of each bin, namely the LDA score, represents the effect size. That is the extent to which a biomarker can explain the differentiating phenotypes among different groups. And in the cladogram, the circles radiating from inner side to outer side represents taxonomic rank from phylum to genus. And each circle here stands for a distinct taxon at corresponding taxonomic rank. The diameter of each circle represents proportionally the relative abundance of each taxon. The yellow knots here stand for taxons with non-significant differences, and the taxons or biomarkers with significant differences will be colored according to their group color. For example, the red nodes here means this microbiota contributes a lot in the group covered by the red color. And letters above the circle and corresponding species are annotated on the right side here. So up to now, we have finished our the analysis showcase. And during communication with our customer, we found that some of them may get confused of whether choosing amplicon sequencing or shotgun metagenomic sequencing to study the composition of microbiota. Here we have a slide showing some differences between them. Firstly, in amplicon sequencing, we will amplify a certain target region. For example, 16S V4. This is our region of interest. While in metagenomics, we do not do any amplification to genomic DNA. We just use them directly to proceed to library preparation. And secondly, the sequencing strategy of Amplicon is paired at 250 BP, while for metagenomics is paired at 150 BP. And we need relatively smaller data amount in Amplicons. Commonly, it's 30K to 100K raw reads per sample. But for metagenomics, often we will sequence a large amount of data. Normally, we choose 6 gigs package. And for Amplicon analysis, we focus on the microbial community structure or composition by doing OTU clustering and diversity analysis. While in metagenomics, although we could also study the composition, what we care more is the genome and its function by doing the gene prediction and annotation based on the various databases. And here we made a cartoon that shows the comparison among resequencing, shotgun metagenomics, and Amplicon metagenomic sequencing, which are the three most common sequencing services you can find on the market. And after we done sampling from like water or feces or soil or gut samples, if we want to study a single bacterium or fungus, so we will do culture and separation. And the most common goals are studying the genome composition, single nucleotide polymorphism, which is the SNP, insertion or deletion, which is the indel, and structure variation, SV. I put parentheses over copy number variation, CNV here is because the structural genes of bacteria are always single copy. So for the mutation research of known microbes, we often choose this microbial resequencing. And if we want to study the microbial diversity at the same time, we care more on the metabolism pathways of genes and gene functions. We just use this total DNA. Of course, it's after host depletion when we will do the shotgun metagenomic sequencing. So if all we care is species identification and diversity, so commonly we will choose Amplicon sequencing, and is also the most cost-effective approach. There are some times that we care more about the whole 16S rDNA than just a few hypervariable regions. This often happens when our purpose is to study gene distribution along the entire 16S rDNA. So in this case, the long read length of a PacBio platform can perfectly fit in. It is able to sequence up to 1,500 BP CCS reads without fragmentation of genomic DNA. And since it's in CCS mode, the PacBio sequencer will sequence a read for a great many times and pick the most accurate one after comparing them to each other. So the accuracy is very high, which can go up to 99.999%. This is a major advantage when we want to identify or quantify some very rare variants. Also, full-length 16S Amplicon in PacBio platform can greatly improve the resolution, that is the percentage of sequences which are classifiable at species level. And this is a statistical figure from a paper, and the x-axis are sample names, while the y-axis is the percentage of sequences classifiable at species level. And it is obvious that the percentage vary from 32% to 93% in Illumina platforms depending on the hyperviable regions, while PacBio, which is shown in the orange bars here, PacBio exceeds totally at almost 100% in each sample tested. So this means by using PacBio, we can get the potential to identify more species or biomarkers than by using NGS platforms. So we are able to obtain a more authentic structure of the microbiota. And at Novogene, we also provide full-length 16S amplicon sequencing service. If you are interested, please feel free to ask our sales representatives for more information. On this slide, we show the workflow and extraction kits recommended for doing full-length 16S amplicon sequencing. The difference between full-length amplicon and NGS amplicon is that we do a full-length 16S rDNA amplification, and we prepare the smart bio library since we use a PacBio platform as a sequencer. And for the analysis part, the most up-to-date GIM2 bioinformatic platform is used to do analysis, which will provide a more accurate and all-rounded analysis to your samples for full-length 16S analysis. So with all that being said, what makes us different from other companies in the market? Well, the most important one is that we have a vast majority of experiences in amplicon sequencing, and our clients had published various papers on related journals. And up to the end of 2019, the total count of published papers by our customers have reached 100, with a total IF of 620. And I'm very proud to say that Novogene has Falcon, a fully automatic intelligent platform that could integrate all pre-sequencing processes from extraction to a library quality control. And in this way, Falcon greatly shortened the project turnaround time by processing 2,000 to 3,000 samples in just 24 hours, which could lead to a 60% decrease of production time. And here we include an introduction video. We will send you this presentation slides. You could click on this blue box to watch. And also thanks to our world leading computing and server capabilities, we could carry a vast number of projects in parallel, and at the same time still achieving amazing data stability and turnaround time. And also, we are very proud that we have more than 2,600 of collaborated research organizations and universities, more than 1,200 collaborated companies, and more than 500 hospitals all around the world. We are proud of so many partners working and doing research with us, and we hope you could be one too. So this brings us to the end of the presentation. Thank you for joining us. And also I would like to thank our fantastic panelist group, Justin, Daphne, Harsh, CB, and Guanhui for supporting today's webinar. And if you like more information, please do not hesitate to see our website at en.novogene.com or send us email at inquiry_us@novogene.com. And for customers who speak Spanish or Portuguese, we have a dedicated sales manager, Dr. Diego Levi, who is now based in Argentina. So please contact us for any information. We are responsive and happy to help at any times. So now I'm more than happy to take questions. You may let us know your questions by the Q&A function in Zoom. And here, the first question we got is from Seth. Are these outputs available from Novogene? Thank you for the question. Yes, absolutely. All the analysis and the data outputs I introduced here, we can do it for you. And another question is that from Sergio, there another option? I'm not very sure. What do you mean by there another option? Do you mean it's if it's I can go back to the slides. Okay, here we have three options for the microbial sequencing. And apart from the Amplicon Metagenomics, we can also do shotgun metagenomics, and the resequencing depends on the research purpose of your project. Okay, let's see another question. And this question is from Magali. Good morning, can we send the DNA samples for sequencing? Yes, absolutely. We accept both DNAs or PCR products or tissues to do this amplicon sequencing. And let's see another one. Here, this question from Michael. Is there any definition about sample number to have enough statistical power? Do you have any material to recommend on this issue? Well, for sample number, I say is basically depends on your project, but we do have some data number that we could recommend for this statistical power. That we have several packages, and from our experiences, we recommend the 100K reads for each sample. That data is enough for our subsequent analysis. And let's take another question. This is from an anonymous attendee that, but it's a very good question. Since PacBio allows for a greater amount of classification, does it use more starting material to obtain this, or is the sample requirement the same for both types of sequencing? And it's definitely not the same for PacBio and Illumina, since you use more in PacBio sequencing. And I can share with you the sample requirements for both PacBio platform and Illumina platform by email after this webinar. And I say another one. And also it's from an anonymous attendee that do you work with Washington University who simultaneously characterize all type of regions in a single assay? I could say that, yes, we work with, I think we work with, okay. I'll answer it later, I think. This is something I cannot answer it here. And another one is, in your experience, is the case in the bacteria. What region is the best to diversity analysis, V3, V4, or V5, as in the case of fungi, ITS or ITS1? Yeah, this is a very good question. So for most of the cases, we can do bacteria identification by sequencing V4, and also it is the most selected region for the majority of our customers. And for fungi, both ITS1 and ITS2 are okay. That depends on your project needs. And you could send us information, and we can do a accurate all-rounded evaluation for you. Okay, and I think that's pretty much it for the Q&A session. And thank you for those wonderful questions. So if your questions were not answered live during this webinar, don't worry. We will answer all questions asked through the Q&A module in the following days by email. So thank you guys for your attention, and I will give it back to CB.
Thank you guys for joining our webinar again. We will be following this webinar up with an email with the link for the webinar, as well as the slides and answers to your questions that were not answered. Thank you again and have a great day. Bye-bye.